Hi guys, today we're doing part 1 of the muscular system, chapter 13, and just prepare yourselves. This is a lot of stuff, but it's very cool, and we're going to talk primarily today about skeletal muscle contraction. I believe this is the first major physiologic process we've talked about where you have to actually know the steps, so just hang in there. This might take a little extra studying. But yeah, I know you guys will rock it. So our question of the day is, how come stiffs are stiff? And stiffs meaning like dead people. So if you are familiar with the term rigor mortis, that's what we're talking about here. And we'll get to that by the end of today's lesson. So let's get into it. For the pre-lecture assignment, this table here should already be filled in from reading the book, but this is a good chart to remember. So know the different types of muscles. We have skeletal, smooth, and cardiac, and then know everything that you have filled in there. So this stuff down here, I don't think you necessarily have to memorize, but this is the most important part of the pre-lecture assignment. Okay, let's look at some anatomy here. So we're going to talk about this going from biggest to smallest. So the biggest part is the muscle belly. And that's what you think of as like your muscle. So you've got your muscles. Let's say this is your part of your bicep muscle in your arm, and it's attached to your bones via tendons, connective tissue. And then you also have this connective tissue wrapped all the way around the muscles in the body. It's just been cut away for this diagram. Let's see. So with the muscle bellies, you usually have one to several that lie parallel. So for example, your biceps are 2 muscle bellies. That's why they're called bi for 2. Your triceps have 3 muscle bellies, which is tri for 3. And let's label tendon here. It's the connective tissue that attaches muscle to bone. And you'll kind of notice here that this is like circles inside circles inside circles inside circles, from the biggest one being the muscle belly. The next biggest one is a fascicle, which you're looking at here. So this is a bundle of skeletal muscle fibers surrounded by connective tissue. So you can see we've got our muscle belly here, which is made up of a whole bunch of these fascicles. And then next we've got our muscle fiber. And when we say muscle fiber, we're referring to muscle cells. So these are just like any other cells in your body. There are organelles, there's mitochondria, there's a nucleus. And then within the muscle fiber or muscle cell, we have what's called a myofibril. And this is the actual contractive element of the muscles. So the contraction, happens here in the myofibril. When that contracts, the muscle fiber or muscle cell is going to contract, which will cause the fascicle to contract, and then the whole muscle belly to contract. All right, so let's kind of write this out in words. Whoops. A group of myofibrils, these guys right here, is a muscle fiber or muscle cell. A group of these muscle fibers here form a fascicle. And then a whole bunch of those fascicles together form a muscle belly. So mainly what we're focusing on today is going to be this little guy, the myofibril, and how it contracts, which will again cause all these other things to contract, resulting in muscle contraction. Okay, so let's talk about the structure of those little skeletal muscle fibers. So anytime you hear the word sarco, it's referring to muscle or skin tissue, so you're going to hear that word a lot today. The sarcolemma is the muscle fiber, so the cell's outer membrane. So we know all cells have a cell membrane. The sarcolemma is what we call the muscle fiber's cell membrane. So here it is kind of peeled away right here. And so we can see inside the cell. And myofibrils contain sarcomeres. And these are bundles of actin and myosin. You're going to hear those two words a lot today. Actin and myosin. So here we have a myofibril, and it's made up of these protein filaments called actin and myosin, which we'll get to in a minute. The sarcomere is where contraction happens. So these will shorten during contraction. So right here, you're looking at one sarcomere, and it's that one element of the muscle that's going to contract, which will, like we said before, cause everything else to contract. The sarcoplasmic reticulum is this kind of blue structure that you see, and it weaves throughout the cell. And this is what stores and secretes calcium. And you'll see why this is important in just a minute. The T-tubules in this picture are green, and what they are is an invagination of the sarcolemma. So kind of the cell membrane making a little tunnel into the cell. And this is how the electrical signal gets into the cell. So invagination of plasma membrane. And transmits electrical signal into the cell. All right, so why are skeletal muscles striated? It's because of these actin and myosin filaments. And if you look at a muscle cell under a microscope, you can see the striations. And that's from the, I'm going to say the longer actin myosin protein filaments. Okay, so now we're just talking about the sarcomere. So we're going to focus in on this little section right here. So again, that sarcomere is made up of myosin and actin. And we call myosin the thick filaments because they are thicker. And we call the actin thin filaments because they are thinner. Finally, names that make sense. During the sarcomere or muscle contraction, the myosin is stationary. So they do not move during contraction. And the actin is what is going to do the moving. It will slide inward during contraction. So here we have one sarcomere from here to here. This looks like a microscopic image possibly, and then we have some models here. So the actin is this red that you see here, the thinner filaments, and the myosin is the thick filaments. And when the actin slides past the myosin, it causes the whole sarcomere to contract. You can see it got shorter from here to here. And we're going to talk about every step of this glorious process. All right, step 1. We got to get the action potential to the skeletal muscle cell. So the action potential is conducted from the nervous system to a motor neuron. So that's nothing new. We know that the effectors are typically muscles, and the message comes from the efferent message comes from the nervous system through a motor neuron to the effector, which in this case is a skeletal muscle. And that motor neuron is going to secrete a type of neurotransmitter called acetylcholine. And then acetylcholine will bind to receptors on the sarcolemma. Remember, that's the cell membrane of the muscle fiber or muscle cell. So this picture is so tiny I can't even really see it, but it looks like we've got the axon terminal here of a motor neuron, and you have these synaptic vesicles which will release that acetylcholine. And let's see, here we've got that acetylcholine attaching to receptors on the sarcolemma, on the cell membrane. So that's step one, get the neurotransmitter to the cell. Through an action potential. Step two, we got to have an action potential on the cell surface that moves into the cell's interior. Oh, before we go from this step right here. So if you remember the curare chemical, and my brain is not thinking of the right word for it, that we talked about, I think in the nervous system, where they would put it on the end of their poison darts or poison arrows, and it causes you to become paralyzed, so you can't move, but you can still feel everything. And so what that drug does is it competes with acetylcholine for these binding sites. So it will block the binding sites, and if that were the case, what's going to happen? Your muscles aren't going to contract, which is why you become paralyzed. But it's not doing anything to your nerve cells, so you can still feel everything. Okay, on to step two, action potential from the cell surface to inside the cell. The action potential travels along the sarcolemma and travels down into the membrane through a T-tubule, one of those invaginations of the cell membrane that we talked about. This is going to trigger the release of calcium that has been stored in the sarcoplasmic reticulum. So here we have the sarcolemma, we've got a T-tubule going down here, and we've got the sarcoplasmic reticulum here that is storing all this calcium, which will then get released into the cell. So the calcium ions are going to flood the interior of the cell and find sarcomeres, which were those little contractile units that we talked about. All right, so we're looking at actin right here, the thin filament. And on those actin filaments is a protein called tropomyosin. That covers the myosin, which is the thick filament's binding sites. And troponin is a protein bound to tropomyosin. Okay, so we've got actin, the thin filament. This green here is the tropomyosin, and I don't know if you can tell in this picture, but there's these little binding sites right here that myosin wants to bind to, and it's covering it up. So right now, in this picture, myosin cannot bind to the actin. So what needs to happen is calcium is going to bind to troponin, this blue right here, which pulls the tropomyosin away, exposing the myosin binding sites. So think of the troponin as a bodyguard for these myosin binding sites, and a bunch of calcium comes along and it gets distracted by the calcium, stops doing its job, and now the tropomyosin moves off the binding site. And now let's see what happens next. Step 4, now the myosin heads bind to the actin filaments, because we have these open binding sites now, to form what we call a cross bridge. Because now we've made a connection between the myosin here and the actin. Step 5, the myosin pulls inwards. Oh, it filled in the pulls actin inwards in what we call a power stroke. So it binds to the actin, the myosin head, and it kind of does this oar-like motion where it pushes back. We call it a power stroke, and it's going to pull the actin in this direction. Step 6. Of course, we've got to have good old ATP here. Remember, that's adenosine triphosphate, the energy currency that cells use. And it binds to the myosin head, which will cause it to detach from the actin filament. So that myosin head has attached here, it's done its power stroke, and then ATP kind of resets it back so it can do it again. So remember ATP, just a quick review of what that was. I'm going to leave myself enough space here. It's adenosine attached to 3 phosphates. And remember that third bond right here is a high energy bond. And so when that's broken, it releases energy that the cell can use. So with that ATP, breaking that bond to form ADP, adenosine diphosphate, because now we only have 2 phosphates, gives energy to cock the myosin head into power stroke position, and the head reattaches to actin to start the cycle again. So it doesn't take just one of these power strokes to contract the whole sarcomere. It's going to take a whole bunch of them, and so the heads have to recock and be able to go again. Okay, step 8. Here's our sarcomere again. So we're kind of zoomed out a little bit. The sarcomere will continue to shorten as long as the motor neuron sends action potentials. And when those action potentials stop, calcium is actively transported, so it's going to be taken back into the sarcoplasmic reticulum, which is where all of the calcium is stored. And now tropomyosin covers the myosin binding sites, and the sarcomere relaxes. Okay, so now troponin isn't distracted by the calcium anymore, so it's going to go back to its bodyguard position and pulling the tropomyosin back onto these myosin binding sites, causing it to relax. Okay, that was a lot, and it's a lot of words and probably a lot of unfamiliar words. So my recommendation would be to get like a scratch paper or a whiteboard or something like that and see if you can write down like step for step in general what's going on. Because you're probably going to be asked what happens after calcium binds to the troponin, or think like you're going to need to know all the nitty-gritty of these steps. I would also recommend, there are some really great videos on YouTube that show this process happening, and there's only so much I can do here just with my paper and my notes. So I would check some of those videos out. Just go to YouTube and type in muscle fiber contraction and watch some of those videos. But be careful because some of them go way more into depth than even this does, and so you don't want to confuse yourself. But I would try to watch some animations of this. And then it asks, back to our question of the day, how come stiffs are stiff? So how come bodies that have been dead for a certain period of time are stiff? And that is because ATP is required to detach myosin from actin. So when somebody dies, they're not breathing anymore, so you don't have oxygen to help you make ATP through the process of cellular respiration, being the final electron acceptor. I'm going way, way back. So without ATP, the myosin heads are going to be stuck in the cocked or uncocked, in the bent position. So the sarcomeres are contracted and they can't let go because you don't have ATP to cause it to recock. Yeah, so cocked position. And the body will stay that way until it's been dead long enough that all of these elements start to decompose, and then that will go away. So yeah, fun stuff to talk about, right? Dead people. Okay, that's it for today. This was a short one. Like I said, make sure you know this table. Just have a quick, maybe do some flashcards with that, and then know those 9 steps that we just went through. As always, message me on Canvas with questions, and I hope you guys have a great day.